Hello, my name is Joshua Mugabe. I welcome you to Genetic Modification and Biotechnology. Our lesson objectives today will be PCR, polymerase chain reaction, GAL electrophoresis. We shall look at DNA profiling, gene transfer, ethics in GMO, cloning, and then stem cuttings. You are welcome. Please do not forget to subscribe. Genetic modification. Polymerase chain reaction. The polymerase chain reaction is an artificial method of replication used to rapidly amplify DNA sequence. So polymerase chain reaction, here we are using this technique to amplify, to increase the sequence of DNA. PCR, polymerase chain reaction, involves three key steps, which are repeated. The first step is denaturation. Here, heat separates the strands, hence no helicase. The second step is annealing. Primers are designated, copying the sequence. And the third step is elongation. Tag polymerase copies the sequence. So here what we are doing, we want to amplify the DNA. If we have small particles or small portions of DNA and we want to amplify them, so what we do here, we use the polymerase chain reaction, and this one involves the denaturation. Here what happens in denaturation, we provide a lot of heat, and when a lot of heat is provided to the DNA, it is going to cause the strands to separate. So the strands now will separate themselves when we have heated it, what we are calling denaturation. Heated it up to 95 degrees Celsius. When the strands have separated, now we go to the second stage, which is annealing. Annealing, what happens is that we reduce the temperatures to lower temperatures, that is around 55 degrees Celsius. And this is to allow the primers. We are going to allow the primers to designate the copying sequence. The primers will come and align themselves on the sequence, on the point which are supposed to be copied. They will align themselves where the copying is supposed to start. Then after that one, we shall raise the temperature a little bit higher to 72 degrees Celsius. And when the temperature is raised, then we are going to the elongation stage. Here, when we raise the temperature to 72, we allow the tag polymerase enzyme to work. Because this one is a little bit heat resistant, it will now copy the sequence. You will see it copying the sequence of the DNA, and then we shall make the copies. A polymerase chain reaction of 30 cycles generates approximately 1 billion copies of DNA. So we have seen the 3 stages. The first one, we said it was denaturation, and during denaturation we've talked about raising the temperature to around 95 degrees Celsius. This one is to help to separate the strands. Then after the strands are separated, we went to the second step, which was the annealing. This one we reduce the temperature to 55 degrees Celsius. And when we reduce the temperature, we are allowing the primers. We are allowing our primers so that they can come and designate or come and position themselves to the points where copying is supposed to start from. That is by lowering the temperature at 55 degrees Celsius. Then the third step is the elongation step. Here we are using our enzyme tag polymerase, which comes, and for it is a little bit heat resistant. We raise the temperature to 72 degrees Celsius. When we raise the temperature to 72 degrees Celsius, this one is going now to force the polymerase, tag polymerase, to start now copying the sequence of the DNA. Now, because we want to amplify this DNA, we can put it in a machine, a cycle machine, which is going to repeat these cycles, denature, annul, and elongate. It can copy those temperatures and use those temperatures, and then it repeats these cycles. So when this machine makes like 30 cycles, those 30 cycles, after those 30 cycles, we would have generated over 1 billion copies of DNA. Thereby, we would have amplified our DNA. So, polymerase chain reaction can be used to amplify. It is used to amplify small amount of DNA. It is used for amplification of the small amount of DNA. Then we have another technique, which is Gale electrophoresis. Gale electrophoresis. Now, in this one, the Gale electrophoresis, this one separates proteins or the fragments of DNA according to their size. It is a technique used to separate proteins or the fragments of DNA according to their size. The latter used for reference. This one is used for reference. The fragments are placed in a gel. The fragments are placed in a gel that is subjected to electrical charge. So we are going to get these fragments that we want to separate. If they can be proteins, they can be fragments of DNA, we shall put them in a gel and then subject them to the electric charge. So the fragments will move to the opposite terminus. They will move to the opposite terminus. Those are the fragments. Now the DNA fragments are placed in agarose gel. DNA fragments can be placed in agarose gel. Remember, we are having very many fragments, different sizes. Now we want to separate them, and we are going to use this technique of gel electrophoresis. So we get these fragments of DNA and we put them in agarose gel. The proteins for them, they are placed in polyacrylamide gel. They are put in polyacrylamide gel. DNA fragments, agarose gel. Protein fragments, polyacrylamide gel. Then the large fragments, those large fragments, of course, for them, they move slower compared to the smaller fragments. Now, the fragments move slower. Those are the large fragments. When they are moving, you will see that the smaller fragments will move faster, then the medium will be in the middle, and then the large fragments will move very slow because they are being impeded by the gel matrix. So that's how now we can separate all those fragments, DNA fragments or proteins. We can separate them, that these are the small fragments, these are medium fragments, and these are the large fragments. The gel electrophoresis is used to separate proteins or fragments of DNA according to their sizes. DNA profiling. DNA profiling. We have the non-coding satellite DNA. Non-coding satellite DNA, and this is composed of recurring elements. The non-coding satellite DNA is composed of recurring elements, and these ones we call them short tandem repeats. They are short tandem repeats, and these short tandem repeats, or STR, they are different for each individual. For example, if we look at our individuals here, we have this male here, we have A, G, A, T. These are repeats, okay? Adenine, guanine, adenine, thymine. They are repeats. Now these ones are 7. 1, 2, 3, 4, 5, 6, 7. Those are the repeats, the ones that we are calling the short tandem repeats. Now again, they will repeat after a certain section. You will find that you are finding again the STR. Here now they are 6. 1, 2, 3, 4, 5, 6. So we can use those STRs to compare the DNA of these two individuals. Now we have our lady here. The lady here has got the STRs. Here they are 4, 1, 2, 3, 4. And then here they repeat, they are 19 of them. So those ones are the ones that we are calling the non-coding satellite DNA, which is composed of recurring elements, which we call the short tandem repeats. So the length of these repeats, the short tandem repeats, will differ according to the individual. So the length is going to differ according to any individual. So when you look at this one, the gentleman, you will see that the length of the first STR is long, and the second, for the lady, the first STR is short. But when you look at the second for the man, the second for the man is short. And if you look at the second for the lady, it is a long STR. So when you are looking at the DNA, you can compare those short tandem repeats, and then you can identify which individual you are looking for. So unique DNA profiles can therefore be established by comparing the STR losses. So on the DNA, now we are going to use the short tandem repeats locally. So we shall look at those locus, and then we can get the different DNA profiles. You look at the different locus, and then you look at the length of the STRs, and then now it becomes easier to identify the individual. So it is unique. It is unique in different people. Unique DNA profile, therefore, can be established. DNA profiling involves comparison of DNA. When we are carrying out DNA profiling, what we are doing, we are comparing the DNA. DNA can be used in forensic. DNA profile can be used in forensic investigations. First is what is happening. DNA sample is collected. Then the STR locus are selected. That is the short tandem repeats. They are selected. Then we can use the polymerase chain reaction amplification. Can use chain polymerase amplification. And then we can use gel electrophoresis to separate them, the fragments. That is in DNA profiling. DNA profiling, we use those techniques. DNA sample is collected. You get the short tandem repeats. Loci are selected. And then you use the polymerase chain reaction amplification. And then we use the gel electrophoresis. DNA profiling in forensic. DNA profiling can be used in forensic investigation. We can use DNA profiling in forensic investigation. Now here, when you look at this profile here, you can see we have a victim. Victim is here. And then this is the DNA that was found at the crime scene. So the DNA of the victim is there. We have our DNA of the victim. This one is the DNA of the victim. And this one is the DNA of the suspect, because it was found on the crime scene. Now, the suspect and the sample should be a complete match. The suspect and the sample should be a complete match. Now we are going to look at our crime scene. This is the DNA of the crime scene, and we are going to compare it with the 3 suspects that we have here. Suspect 1, suspect 2, suspect 3. We want to compare their DNA and we see who is the suspect. I'd like you to compare those DNA profile of suspect 1, suspect 2, suspect 3 with the crime scene DNA. So when you look at that profile, you will see that the suspect is suspect 2. The profile that is found at the crime scene is the same as that one, the DNA profile of suspect 2. So that's how DNA profile can be used in forensic. DNA profile can also be used in paternity tests. The DNA profile can be used also in paternity tests. For example, we have a mother here. We have our mother here. The mother has produced a baby, but there are 3 possible dads. There are 3 possible dads, and we need to find out who is the father. So we are going to look at the profile of the mother, DNA profile of the mother. We shall also compare it with the profile of the child, and then we shall compare them with the profile of the dads. Because children inherit DNA from both the parents, all of the children's bands should match to either the father or the mother. That is combination of the parents. The baby's DNA profile is a combination of the parents. So there should be a match to the mother and the father. So now here we shall start matching. This one is the profile for the mother, and this one is the profile for the baby. The profile for the mother should match with the profile for the baby, and the profile of the baby should also match with the profile of the father. So when we look at it, if we start matching, what we are going to see is that now this one here matches with the mother, and this one. And even this one here matches with that one of the mother. Even this one matches that one of the mother. Then we go now and look at the remaining ones. The remaining ones should match with that one of the father. We start with this one of the baby. If you look at this one of the baby, whom does it match with? It is likely to match with this one and this one. Okay? Then we go to another one. We go to this one of the baby. Whom does it match with? Now it comes and matches with this one. But again, it also matches with this one. Then we go to this other one for the baby. If you look at that one, it matches with this one, and then also matches with this one. So now when you look at the matches, you start now counting who is the probable father. This one has only one match. That is Daddy 1. Daddy 2 has 2 matches, okay, with the baby. Daddy 3 has 3 matches if you look at that one, this and these 2. So the probable father is that one who is parent 3 with this DNA profile. So this one is the father of the baby because he matches more compared to the other. Gene transfer. Gene transfer. Gene transfer is the process by which genes from one organism are inserted into another organisms. Here we want to transfer the genes, and we are transferring them. These genes, we get them from one organism, and then we insert them into another organism. That one is what we refer to as horizontal gene transfer. Now, genetic modification is carried out by gene transfer between species. Genetic modification is carried out by transfer of genes between species. So the gene transfer involves 4 key steps. If we are transfer the gene, it will involve 4 steps. 1. We shall have DNA extraction. Here we shall get the gene of interest and then also the vector. Or it can be a virus or a plasmid are isolated. Now, here we are going to transfer the gene from one organism to another. But we need that gene that we are interested in, and we need to get a vector which is going to transfer this gene into the host where we want it. And in most cases, the vectors that are always available are the viruses and the plasmid. Now, the second stage is the digestion. The gene and the vector are cut with restriction endonucleosis, with restriction endonuclease. Endonuclease is an enzyme. So it is the one now that is going to cut that gene of interest, the gene that we are interested in to transfer. We shall use restriction endonuclease enzyme to go and cut that gene where it is located. And then we shall also use the same enzyme to go and cut on the vector, to cut that point where we want to insert the gene. It can be a plasmid. It will go and cut the plasmid and then where exactly we want to place that gene. That's what we are calling the digestion stage. Then there is the ligation. The ligation stage. The gene of interest is placed into the vector. Ligation, this one is a stage where we get the gene of interest that we have cut from our source, and then we are placing it into the vector where we want it to be transferred from, or by, or with. Then we have the transformation. Transformation, now the recombinant vector is inserted into the host cell. Now we have already fixed our gene of interest into the vector. And what we form after fixing the gene into the vector is what we are referring to as the recombinant vector. Now, transformation is now the transfer or the insertion of this recombinant vector into the host cells. We are inserting it in the host cell in another organism. Gene transfer vector. A vector is a molecular vehicle used to artificially carry the genetic material into a cell. So our vector is the one which is going to help us to transfer this gene of interest into another cell. So we call it a molecular vehicle. It is going to help us artificially to carry this genetic material into the cell where we want to take this gene. That is what we call a vector. Now, we have a plasmid. We say that the most useful vectors that we have viruses and plasmid. We have a plasmid. A plasmid is a circular DNA molecule, and it is capable of autonomous gene expression. It can express freely itself, okay? So here we are going to get our plasmid. And this plasmid, we are going to have the insert. This is what we are going to insert. We are going to insert our gene here. That's where we are going to insert our gene at that particular point where we are going to cut. And then it has also got what we call the promoter, and it has what we call the selection mark. Okay, but that is where we are going to put our, in the red, insert. That's where we are inserting our gene, which is going to be transported. Then there is also the use of the viruses. The virus, a virus can inject their genetic material directly into the host cell. So the virus can also be used to carry its genetic material directly into the host cell. But this one is not mainly commonly used because you don't know how the material which is carried inside, how it will behave inside there in the host cell. So we end up using the plasmid. Restriction enzyme. Now, the restriction enzymes, they cut at a specific recognition site, and they create blunt or sticky ends. Now we say that we use the restriction enzyme. We use the restriction enzyme, if you remember very well, endonuclease enzyme, to cut the specific gene of interest. And it is also the same enzyme we are going to use to cut the specific position on the plasmid where we want to insert the gene. So it may create a blunt position where we want to cut. It may cut it as blunt like that, and it cuts the specific gene that we want, or where we are going to place our gene. Or it may cut it as a sticky end. Now when it cuts it as a sticky end like this, that means that now the other one which is going to come, the gene that we are going to bring, should be complementary with these bases. It should fit in those particular bases. The gene that we are going to bring should fit in those particular bases where they have been cut. This one is the sticky end. This one is the blunt end. The blunt end, it just fits inside. But in the sticky end, it has to fit at those particular bases. That is the restriction enzyme. It cuts a specific recognition site where we want to insert our gene. Gene transfer. We have DNA ligase. Now, once the gene and the plasmids have been cut with restriction enzymes, they are joined to form a recombinant construct. So what we have said, we are using the restriction enzyme. The restriction enzyme is the one that cuts at the plasmid at that particular point and that particular point. And we shall use still the restriction enzyme to cut the gene at that particular point and that particular point. So when you look at this type of cutting, this is not a blunt cut. This is a sticky cut. So it has cut the vector at that particular point, and it has also cut the gene at that particular point. So here we shall now use DNA ligase. DNA ligase is an enzyme which is going to help us to combine our gene with the vector. The gene is the one that we are referring to as the insert. It is the one which is going to combine them. And when they have been combined, the gene and the plasmid or the vector, now we refer to them as recombinant plasmid. They are referred now to as recombinant plasmid because they have been joined together by the DNA ligase enzyme. Now, the fusion of the gene and the vector is mediated by DNA ligase. Our DNA ligase is the one which mediates the fusion of the gene and the vector to form a recombinant plasmid. The DNA ligase joins together the backbones of the gene and the plasmid by forming a covalent phosphodiester bond. So the DNA ligase is going to join the plasmid with the gene, and the bonds that they are going to use are what we are referring to as phosphodiester bonds. The recombinant constructs can be identified and isolated via gel electrophoresis. So we have now our recombinant construct. The recombinant construct is the recombinant plasmid, the one which is made up of the plasmid joined with the gene. It's what we are referring to as recombinant construct. So that one can be identified and isolated via gel electrophoresis, whereby this one will be larger than the unincorporated plasmid. So our recombinant construct is going to be larger when you compare it with another plasmid which has not been incorporated with the gene. So our recombinant is going to be bigger because we have already inserted inside our gene. So it will be bigger than the unincorporated plasmid. Please don't forget to subscribe. Gene transfer, gene delivery. How are we going to transfer the genes? The recombinant construct is introduced into the cell to be genetically modified. So we are transferring our recombinant construct into another cell to be genetically modified. Introducing foreign body results in a cells being considered transgenic. So when we transfer the foreign DNA and transfer it into another cell, now that cell where we have transferred it, now we refer to it as transgenic. We call it transgenic cell or transgenic organism. The introduction of the construct can involve either one, transformation. In transformation, now the plasmid is inserted in the bacterial host cell. When we get our construct and we transfer it in the bacterial cell, host cell, that type of introduction is called transformation. Transformation, the plasmid is inserted in the bacterial host cells. Then another one is transfection. In transfection, the plasmid is inserted into eukaryotic host cell. When we transfer the plasmid and put it in eukaryotic host cells, that one is what we call transfection. Transfection. Eukaryotic cells, these are cells that have got a nucleus. They have got membrane-bound organelles. They are what we call eukaryotes. So when we transfer that plasmid or our construct we have made and transfer it in eukaryotic host cells, that type of transformation is what we refer to as transfection. Then we have another one we call transduction. In transduction, the viral vector is used for delivery of DNA, foreign DNA. Now here in transduction, we are using now the vector as a viral vector. Our vector we are using to transfer the gene can be a virus. Therefore, that one is referred to as transduction. So those are the ways how we can introduce the construct into the cells. Transformation, we are putting the plasmid in the bacterial host cell. Transfection, we are putting the plasmid in the eukaryotic host cells. And then transduction, we are using the viral vector to transfer our foreign DNA. There are a variety of techniques that we can be used to introduce construct to the cells. This includes electroporation. We can use microinjections. We can use biolistics and liposomes. Those are other variety of techniques that can be used to introduce the constructs to the cells. Selection and expression, still on gene transfer. Now, antibiotic selection may be used to identify and isolate the transgenic cells. Now, remember, the cells where we have put our construct, those cells we are saying we call them now transgenic cells. Those are the cells where we have put our gene or the plasmid. So now how can we isolate the transgenic cells from other types of cells where we have not placed the plasmid and the gene? So here we use antibiotic selection. The antibiotic selections will help us to identify and isolate the transgenic cells. Here we have our antibiotics. The antibiotics, of course, they will prevent the growth. But what is going to happen is that the antibiotics will prevent other cells from growing. But the transgenics, those are the ones where we have placed our plasmid, they are resistant to antibiotics. For them, they are resistant to antibiotics. So what is going to happen for them? They will be seen growing because they are resistant to antibiotics. The other cells where we have not placed our plasmids, they are not resistant to antibiotics. So that one will help us to identify and isolate the transgenics. The plasmid vector contains antibiotic resistance gene. That is the plasmid vector. It contains antibiotic resistance gene. So only cells that have incorporated with the plasmid will grow in the presence of antibiotics. It is only the plasmid vector that contains antibiotic resistant gene. So the other cells which are not incorporated with the plasmid, they will not grow. So the only one which is going to be resistant to the antibiotics, it is the cell which has been incorporated with the plasmid. Gene transfer overview. An example of gene transfer is the mass production of human insulin by bacteria. For example, here, one of the examples where we use gene transfer is in the production of human insulin. Human insulin is used to treat diabetes. Now, we get the human cell and we get our gene that we are interested in, the one for insulin production. And then we also get the bacteria. When we get the bacteria, we shall remove the plasmid. And the plasmid is where we are going to insert our gene that we have got from the human cell. Now we shall incorporate them together like the procedures that we have seen earlier, and then we shall form our recombinant. Our gene has been incorporated into the plasmid to form a recombinant plasmid. Then we shall transfer this recombinant plasmid into a bacteria. When we transfer that plasmid into the bacteria, now this bacteria becomes transgenic bacteria. Now it can be grown in the culture, and then now we can extract our insulin, which will be used. Bacterial cells divide rapidly by binary fission, and so can produce large yields of proteins in a short period of time. and human insulin is used to treat type 1 diabetes. So we are now using the bacterial cells to produce for us insulin, which we are going to use. Why? Because bacteria divide rapidly by binary fission, and therefore they can produce large yields of proteins within a shorter period of time. And those proteins, the insulin, is the one that we are going to use to treat type 1 diabetes. Ethics of transgenics. The benefits of genetically modified crops. Benefits of genetically modified crops. 1. They are for nutrition value of food, meaning that the nutrition value of food could be improved. 2. Crops could be engineered for increased yield. We can get crops that can yield too much. And then crops would have a longer life share. We can have crops that have longer shelf life that we can keep for a longer period of time without going bad, like tomatoes. Then crops could grow in a wider range of conditions. We can form crops that can grow in a wider range of conditions. Insect-resistant crops would reduce reliance on pesticide. So if we modify and we get insect-resistant crops, then that one will help us to reduce the reliance on pesticides. Potentially reducing farming costs and carbon footprint. So use of transgenics or genetically modified crop can help us to reduce the farming cost. For example, we are not using pesticides, we are not using chemicals. Those ones, all of them now, they are cutting down the cost. Could reduce the need of land clearing because they are high yielding. We have got those ones that are high yielding. We can stop land clearing, and this one will decrease deforestation, because if you have been using a very big piece of land to cultivate a given amount of products, now you can use a smaller piece of land and then you cultivate more products compared to what you would get from a very bigger piece of land. What are the risks of genetically modified crops? 1. New traits could have unknown long-term effect. We don't know what effects they will have in a long term, because certain proteins can trigger allergic reactions. They can trigger allergic reaction, but we don't know that one. Then cross-pollination could lead to super weeds. If they are cross-pollinated, it could lead to weeds that are very difficult to control. Then patents could lead to monopolies over crop use. What we are producing can lead to monopolizing other crops. Crops might impact native species. The crops may reduce the biodiversity of the native species. We cut down the local species and then we plant the genetically modified one. So with the time, the biodiversity may reduce. Different regions might have different GMO standards, and making appropriate GMO use very difficult to police. So you may find that some GMOs are not allowed in certain areas, and this one will be very difficult for the police to control. GMO examples: BT corn. The BT corn is a transgenic crop that produces insecticide from Bacillus thuringiensis. Now, this corn here, which we are calling the BT corn, it is a transgenic crop. And being a transgenic crop, it produces insecticides that are dangerous to toxic to the European coat borer. So the European coat borer, which feeds on it, will of course be destroyed by the plant itself. So there is no need for using pesticides. That's one of it. This insecticide is toxic to the European corn borer that would eat the crop. So the plant itself does not require pesticide to control it from the European corn borers, because it produces the insecticide which is toxic to the borers by itself. There are concern about the BT corn is impacting survival of the monarch butterfly. So the problem now with the BT corn is that now it is impacting the monarch butterfly. The monarch butterfly, it feeds on the milkweed, but the milkweed the monarch butterfly feeds on is dusted with the pollen which is coming from the BT corn. And that pollen which is coming from the BT corn has a negative effect on the monarch butterfly. So the monarch butterfly are reducing because of the pollen which falls on the milkweed, which is eaten by the monarch butterfly. But the pollen is coming from the BT corn. Pollen from the BT corn is dusting the milkweed plant which the butterfly feeds on. Please don't forget to subscribe. GMO example, we are still on the BT corn. Analysis of the risk to monarch butterfly of the BT corn. Butterfly mortality higher when fed plants dusted with BT corn pollen. This is in the lab. When they tried to carry out the testing in the laboratory, it was observed that the survival of the monarch larva went on reducing with days. When there was BT pollen dusting, with days the number of the larvae went on reducing. And when there were no BT pollen grain, it remained constant. So that one. So they say that there is no significant mortality change to the butterflies near the BT in the wild. Now here they carried it out, the test in the wild, and found that there wasn't any change in the butterflies near the BT corn when they were experiment carried out in the wild. So they found out that the survival rate for the larva went on decreasing with the day. That is, the larva near the BT corn, it reduced. And even the test for the larva, when there is no field nearby there is no BT corn around, still it went on reducing. So which means there wasn't any significant mortality change to the butterfly near the BT crops in the wild. Cloning. Clones are a group of genetically identical organisms derived from a single parent cells. The clones, these are groups of genetically identical organisms derived from a single parent cell. Clone can occur naturally, that is via asexual reproduction method. For example, if we cut a stem of a vegetative plant and we plant it, it will give us the same plant with the same genetic makeup. So they will be identical, that is in asexual reproduction. It can occur artificially via embryonic division or somatic cell nuclear transfer. Cloning in animals. Certain species of animals can clone themselves in a variety of ways. This includes binary fission, for example in planaria. Here the organism divides equally into two and each one turns into a new organism. Then we have budding. Budding in Hydra. The cell splits off from the parent organism and it grows into a new organism. Then there is fragmentation. Fragmentation in starfish. New organisms can grow from a fragment. A fragment breaks off and develops into a new organism. And then the other one is reconstructed to form the missing part. That is natural cloning in animals. Natural cloning in plants. Plants have a capacity for vegetative propagation, whereby smaller parts of the plant can be induced to grow independently. This is because plants retain pluripotent tissue. They retain pluripotent tissue, which are the meristems. For example, here you can see a bud. This bud can turn into a leaf, grows into the leaf. And then at the node, it can grow into the roots. Stem cuttings are separated portions of plant stems that can regrow into new independent clones. We have seen, for example, sugar canes, we can use stem cuttings. Cassava, you can use stem cuttings, and many other plants. So stem possesses nodes from which the leaf, root, and branch grow. So those nodes, it's where the stems are going to grow from. It's where also the roots are going to grow from. Various factors may affect the rooting of the stem cutting. Natural cloning is still in human. In humans are capable of creating genetic clones. That is, even humans are capable of creating genetic clones. For example, we have identical twins, whom we refer to as monozygotic. These ones are created when a fertilized egg splits into two separate cell groups. This method of cloning can also occur artificially. This method of cloning can also occur artificially. Separation must occur early. Separation must occur early, and that is during the molar stage. This cloning method is limited by the fact that it still occurs after sexual reproduction has occurred. That was why it is limited. It takes place when fertilization, sexual reproduction has already occurred. Animals can be cloned at embryo stage by breaking up the embryos into more than one group of the cells. Artificial cloning. Artificial cloning. Clones can also be developed artificially using differentiated tissue of adults. Now, this method of cloning is called somatic cell nuclear transfer, SCNT, somatic cell nuclear transfer. This method of artificial cloning can be used for two main purposes. One, reproductive cloning. This one produces new organisms. For example, when we want to repopulate the endangered species. Two, therapeutic cloning. This one produces new tissues that can be used in transplantation or they can be used in disease treatment for therapeutic purposes. Currently, this technique has low success when applied to reproduction. Artificial cloning currently has low success rate when applied to reproduction. Human reproduction cloning is considered unethical and is currently illegal. Human reproductive cloning is considered unethical and is currently illegal. We are not supposed to clone human beings. Artificial cloning, somatic cell nuclear transfer. Somatic cell nuclear transfer involves cloning embryos from differentiated adult tissues. Here, the nucleus is removed from the adult body cell. That is the new nucleus. Then, the egg cell nucleus is destroyed by ultraviolet radiations. The body cell nucleus is then fused in a nucleated egg. Electrical current stimulates the egg to divide. And then developing embryo is implanted into a surrogate. So here what we are looking at, we have an adult organism. We can get the cells from the adult organisms, and these cells, we can extract the nucleus. We remove the nucleus. Then we can get an egg. When we get an egg, we remove its nucleus or destroy its nucleus by using ultraviolet rays. Then afterwards, we get the nucleus that we got, and the egg which we have destroyed its nucleus, we insert in our nucleus which we got from the other cells. And it is now implanted into a surrogate mother. It will be implanted into a surrogate mother. Now it will develop into a new organism. So the embryo develops into a cloned animal. That embryo that we got is going now to develop into a cloned new animal. Thank you very much for participating in this genetic modification And today we have looked at PCR, polymerase chain reaction. We looked at the gel electrophoresis. We talked about DNA profiling. We looked at gene transfer. We talked about the ethics of GMOs. We looked at the cloning. And lastly, we looked at the stem cutting. Thank you. Please don't forget to subscribe.